For the introduction. I'm afraid that I've lured you all here under false pretenses. Ostensibly, I'm supposed to be talking about yuzu, which is my latest fruit, which accelerates in-silico saturation mutagenesis using compressed sensing. However, no matter how hard I tried, I couldn't compress all the details of how that method worked into a 10-minute talk. So instead, what I'm going to be talking about is Crookshanks. Crookshanks is my wife's 20-year-old cat, which she's had since childhood. For those of you in the audience who feel young, that's right, the height of the Harry Potter mania was indeed 20 years ago. We picked up Crookshanks from her parents' house, coincidentally just at the start of the pandemic, which was great timing because I immediately fell in love with him. And like any self-respecting geneticist, might do to a beloved pet or perhaps a significant other, I got him sequenced. The first report that we got back covered coding mutations. And so these mutations are mutations that happen within the genome that influence the influence the sequence of proteins and can potentially lead to disease. Unfortunately, the results that we got back were sort of a mixed bag. On one hand, we weren't able to find any mutations that happen in coding regions that would be interesting in the study. So that was good because it meant that it helped explain why he has lived for so long. But unfortunately, it hasn't explained at all why he is, according to my wife, a truly feral creature. The next thing that we got back were the raw reads. These were the raw reads from the sequencing experiments. I then mapped these to the cat reference genome felcat9. I know that all the cat geneticists that are in the audience are narrowing their eyes at me. Why are you using an Abyssian cat reference in order to study a cat that is clearly a domestic American shorthair? Well, unfortunately, it was the best annotated cat reference that exists out there. What you'll see here is a pile up of the reads. Each one of the horizontal bars corresponds to the mapping of a read to the reference genome. And all of the colored lines that you'll see there that are highlighted correspond to variants, mutations that Crookshanks has compared to the reference cat genome. And let me tell you, Crookshanks is full of mutations. There are just 2 of them here, but even after filtering out ones that aren't really reproducible, there are still over 1 million mutations that he has. So then the question becomes, most of these mutations don't happen in genic regions, so what are they actually doing? A pretty popular approach for trying to understand these is to run the reference sequence and the variant sequence through some sort of predictive model that predicts some sort of aspect of biological activity and seeing what the difference is, whether or not it's interfering with that type. So we can do that with a model here that predicts CTCF activity. This is just predicting, what we're seeing here is a CTCF peak that's predicted from the model using the reference cat sequence. You can see there's sort of 2 peaks. There's one really strong peak followed by a second one that's not as strong. When we put in Crookshanks' sequences, or rather when you put all the mutations, which are the black bars there, you can see that the strong CTCF peak disappears. That suggests that the variants that are in this region correspond to, are responsible for reorganizing chromatin, and so they might be interesting to look at later. But simply looking at the variants that happen within a sequence is not always sufficient. You need to know the context in which they occur. And so commonly, people use an approach called in-silico saturation mutagenesis. What this does is that it first runs your reference sequence through your predictive model, and then it runs all possible single mutated sequences through. So if you have a sequence of length L, you're ultimately running 3 L mutated sequences through as well. You run it through your model, you get the output for your reference sequence, and then through all of your mutations, and then you aggregate those differences in order to get some sort of score. Commonly, you just take the difference, and then you sum up the differences, and the mutations that cause the largest differences are thought to have the highest variance score. But I noticed that this was taking a very long time. Imagine for each locus that you want to investigate, you need to not only run the original sequence through, but you need to run all possible mutations through as well. That can take a very long time. But then I noticed, if you do something like this, you're probably going to see a lot of values that come out after this difference that are exactly 0. And the reason is pretty simple. Imagine that your model is just a simple single convolutional layer. You scan your convolution over the sequence, and then you take this difference. a single mutation from reference can only influence the output of the convolution within the kernel width of the convolution itself. By definition, it can't make a difference outside of that. And so my question was, why are we wasting a huge amount of time running ISM when, by definition, so many of these values have to be 0? So this is where the idea of yuzu comes in. Basically, yuzu operates on the difference between the mutations and the reference sequence as opposed to the actual sequence itself. So what we have here is that we would take the mutated sequence and we just get a tensor that only has a +1 where the mutated sequence is and a -1 where the reference sequence is. We then take that using, oh, it's cut off at the top. Oh well. We multiply, and then we compress all of those mutated sequences using the theory of compressed sensing into a much smaller set of probes, which we then apply the convolution to. Instead of applying our convolution to all 3L sequences, we're applying it to a significantly smaller amount. And that's where the speed up from yuzu comes, that you're applying it to a significantly smaller amount of data, but where each operation is now informative. Every operation gives you something new. So you then apply your convolution, you get your output, and then you can use the theory of compressed sensing in order to decode that on the other side in order to perfectly reconstruct your scores. And I want to say it's perfect. There is mathematic guarantees that what you get out of this is not just, close to what it is, but that it's actually identical. So we then looked at a few common models, and what we have here is the speed that it takes yuzu to decode a bunch of sequences in the bottom compared to naive ISM, which is in purple, and another approach called fast ISM in blue. You can see that yuzu is significantly faster than traditional ISM, frequently an order of magnitude faster, while also faster than fast ISM in most settings, particularly the interactive session, which is particularly important. Then the interactive session is like you have a Jupyter notebook open and you want to see what's going on in one sequence. You don't want it to take forever like you'd see fast ISM would take. So I'd like to thank everyone who was involved in this project. I would encourage you to read the details read the details of yuzu in our bioinformatics paper and also the details of fast ISM in a separate bioinformatics paper. Thank you.